Hello, and welcome back to Introduction to Genetics and Evolution. I'm very excited about this set of videos. What we'll be doing now is tying together the content from various parts of the class. So we'll be talking about things from transmission genetics, quantitative genetics, population genetics, molecular evolution, and tying it all together to talk about how these concepts can be applied to help humanity. So let's get started. Just to put it out there for you, these insights are very important. These are things for which there are direct human medical applications and other sorts of applications as well. As I like to say, it's not all just math and dinosaurs, as people from outside the community sometimes tend to think. What I'll do is I'll divide this video into three sections. I'll talk very briefly about quantitative genetics, which obviously has had a huge impact. I'll be repeating some things that you saw earlier. I'll talk briefly about selection and microbes, also repeating some things that you heard earlier. And finally, I'll go into two vignettes, two stories about applications of evolutionary insights. Now, obviously, quantitative genetics has been applied for many, many years, even if people didn't give it that particular term. It's long been known that there are similarities between parents and offspring, and that some traits are more heritable than others. There are some traits for which you will look more like your parents, or you will behave more like your parents, than other traits. Now, this similarity is what's been applied in the context of selective breeding. So this is what farmers have been doing for many years. These are what people who grow livestock have been doing for many years. And importantly, people who are rearing different kinds of pets have been doing for many years. Many of you know, of course, the ancestor of a modern dog was much more wolf-like. But yet today we see this incredible variation. And this variation came about as a result of selective breeding. People identifying traits in particular dogs, getting dogs with similar traits to have kids, and just repeating it over and over again until the trait was more and more elaborated. You had the small size of the Chihuahua, or behaviors of particular kinds of dogs. Darwin was very familiar with this in the context of various, fancy pigeons, and this is something that's been a common thread for people historically. But importantly, look at crops. This is a standard mustard plant here. This is actually the ancestor of many of the foods that you eat all the time, like broccoli, cauliflower, kale, etc. I mean, these are the things that are direct products of artificial selection. So these are, in fact, applying quantitative genetic principles, applying evolution, though in this case guided by humans. Now, antibiotic resistance is an example we talked about earlier, and the example I told you about was that of penicillin, which was first mass-produced in 1943. At the time, it seemed like the wonder drug, and you can see that from this ad over here, where thanks to penicillin, he will come home. Over time, the bacteria, which were initially dying in response to penicillin, stopped doing so. The first resistant staph strains were found in 1947. By 1950, 40% of staph isolates were resistant. By 1960, 80% of staph isolates were resistant. Now, how did this happen? Well, you saw this earlier, but hopefully now you have a better understanding that we had natural selection for bacterial resistance to antibiotics. That is actually still happening. We had this original population. The blue ones were susceptible to antibiotics. The red ones were resistant. The red ones were in the far minority. After the antibiotic was applied, we killed off a lot of the susceptible strains. The resistant ones were the only ones that were left. They had no competition, and they have spread. As a result, a lot of bacteria that are out there now are antibiotic resistant, far more so than before human application of antibiotics. And in that regard, antibiotics were and very often still are overprescribed. Now, I do not advocate stopping all antibiotic usage, because obviously these are things that are helping humanity getting rid of illness and things like that. The thing I would emphasize to all of you who are watching this is to avoid antibiotics as a preventative measure. When your doctor tells you, well, I don't think you really need this antibiotic, well, why don't you take it anyway, just in case? No, that is bad, because what that's doing is that's continuing to select for more resistant strains out there. Wait until you know you need it and then take it, because you may not need it. What's also happened alongside this is that a lot of good microbes have been killed. And we have, in fact, humans have co-adapted with a lot of these good microbes that, again, they synthesize our required vitamin K, they help us to resist invading organisms, and they affect regulation of stomach hormones. But by our over-application of antibiotics, we've killed a lot of the good stuff too. And there was a study in 2011 showing that an increase in risk of inflammatory bowel disease in response to number of courses of antibiotics that kids were given. That those kids with no antibiotics had a very low incidence of inflammatory bowel disease, and it went up with more and more courses. Now, again, these are stories you already saw, so let me tell you a couple of new stories. First, I'd like to tell you about the story of the control of mosquito spread of dengue using intracellular bacteria. This was a really cool case where an evolutionary insight may actually save a huge fraction of the world's population. And next, we'll talk about the human evolution of lactose tolerance, or lactase persistence. So let's start with dengue. So let's start by looking at dengue. Dengue is obviously a bad disease. It is caused by a virus, which is spread through mosquitoes. It is, in fact, the leading cause of childhood death in Southeast Asia. And in fact, not just Southeast Asia, but 40% of the world is at risk for infection from dengue. There have been epidemics in various parts of the world, including here in the Americas, as recent as 2009. And they'll probably continue to happen for quite some time. So how do you control this disease? Well, two options have been tried. One is to kill mosquitoes, basically go in there and just wipe out as many as possible. That helps, but the problem is we can't get rid of all the mosquitoes out there. The other is to avoid mosquitoes, things like nets, things like that, to basically keep humans away from them. Again, that has some effectiveness, but it doesn't eliminate the problem. Unfortunately, there's no available vaccine yet. So let's set that aside. That's what's happening in terms of dengue. Now let's talk about an evolutionary study. What's happened completely separately from what was going on with dengue is research on this Wolbachia bacteria. This is an intracellular parasite. It's a parasite within insect cells. It's extremely common across insect species. Maybe 80% or more of insect species have Wolbachia in them. It's passed through the eggs, so it's totally maternally transmitted. So, mothers pass it to the kids through the eggs. Interestingly, there's often this pattern called cytoplasmic incompatibility associated with it. That basically you get no offspring when you mate an infected male to an uninfected female. So infected male to infected female is fine. Uninfected to uninfected is obviously fine. But interesting, if you have an infected male and an uninfected female, so I have here's the male symbol, infected male and uninfected female yields no offspring. All the others yield offspring. So let's think about what this does. This creates a very interesting evolutionary dynamic. Now, so looking at this chart, let's focus on the columns here. Is it better? Now, if you imagine some of the females in the population are infected and some are uninfected. Some of the males in the population are infected and some are uninfected. Is it better as a female to be infected, or is it better to be uninfected? Think about that for just a second and try this quick question. Well, I hope that was a very easy question for you there. If you're looking at the infected females, they can always have kids. When you look at the uninfected females, they can only have kids if they mate with an uninfected male. So in fact, it's actually better to be infected. As a result of this dynamic, when insects have Wolbachia infections, the infection spreads very rapidly because there's actually an advantage to females who are infected. And since it's only passed maternally, this advantage to females allows it to spread without any problem whatsoever. Now, linking these two things together, it's easy for Wolbachia to spread in insect populations, and we have this dengue fever thing. In 2009, there were a couple of interesting papers. One was published showing that you could introduce slightly life-shortening Wolbachia into mosquitoes. In shortening the lifespan slightly of these mosquitoes, they were less likely to actually transmit dengue fever. That's pretty cool. And you can actually introduce this Wolbachia that makes it so the mosquitoes are still alive, they're still having kids, but they're not actually transmitting dengue. Another study in 2009 showed that Wolbachia directly inhibits viruses from infecting the host. So it's actually potentially blocking them from getting Wolbachia. The Wolbachia infection actually may cause an immune upregulation and viral inhibition, as it was elaborated in an even later study. And so if you introduce Wolbachia into mosquitoes, you make it so those mosquitoes are less likely to give the disease to humans. That's cool. And this Wolbachia infection will spread because of that female advantage to being infected. So what's happened since that time, there actually have been controlled Wolbachia infected mosquito releases in Australia to test this out. So up here in northern Queensland, they released a set of mosquitoes that were infected with Wolbachia, and they looked at what fraction of the mosquitoes captured actually carried Wolbachia. So this is time in days on the x-axis. On the y-axis is percentage of the population that had Wolbachia. And you can see very rapidly from these couple of releases, the entire population was infected with Wolbachia. Again, this is great because in being infected with Wolbachia, you're less likely to transmit dengue. So as a result, there's an evolutionary study that may reduce or potentially even eliminate dengue out there. This was leveraging purely evolutionary research on Wolbachia, and nonetheless, the outcome may affect 40% of the world's population that is at risk for this disease. That is cool, isn't it? So let me tell you another very cool story. Let's look at understanding the human evolution of lactose tolerance. Some of you out there may have experienced this problem of lactose intolerance. Now, when you're lactose intolerant, which my daughter is actually, you get extreme painful gas when you consume milk products. And this typically isn't something that happens right off the bat when you're a baby, but it's something that starts when you're sometime more than 5 years old. Sometimes quite a bit older. Sometimes it starts when you're a teenager or something like that. Now, actually, if you go back in time, if you go back 10,000 years ago, all humans were actually lactose intolerant. The mutant condition is actually tolerance. You may think, well, how did that happen? Well, think about it. How many, 10,000 years ago, how many adults are drinking any sort of milk? None. So there was no cost to this, right? So you were not nursing as an adult, so there was, you obviously didn't need any human milk. We hadn't yet domesticated cows, so there was no problem associated with this. Today, actually, we see quite a bit of variation that, among the Dutch, you see a very low incidence of lactose intolerance. Among European Americans, again, pretty low. African Americans, kind of intermediate. And a fair number of Southeast Asians are actually lactose intolerant. But what happened? What changed all this? Well, it was found that in Europe, about 7,500 years ago, there was a one base pair mutation that happened that allowed production of lactase enzyme. This is the enzyme that breaks down lactose and makes it so you don't have these stomach problems. It breaks down lactose in adults. It allows you to digest lactose. Now, this timing, this 7,500 years ago, coincides with the onset of dairy usage in Europe. So what this did is this allowed, an extra source of nutrition and nourishment for people that were out there. There was an advantage to it when this mutation arose. There was a selective advantage associated with it. If you look at your earlier European fossils and sequence them, they in fact do not have this mutation. So we know this actually happened. Coinciding with what we expect from molecular evolution, there was a selective sweep around this new mutation. I actually made up this particular figure. This isn't true data, but imagine here on the x-axis is position in base pairs. On the y-axis is pi, a measure of nucleotide diversity, or nucleotide variation. If you looked right here near the mutation, this Eurasian CT mutation, which allows lactase persistence, you see much lower pi in the regions further out. So there's a long stretch with no nucleotide variation around lactase in individuals having this new derived tolerant allele. This is clearly not the result of genetic drift. This is something that would be expected from a selective sweep. So that's very cool. I made up this particular data here, but this was just illustrative to show you what happened. So it sounds like this is a cool story, but in fact, the story is more complicated. In fact, there were multiple mutations. In Africa, there was a different one base pair mutation that happened at a different time, in this case 5,000 years ago rather than 7,500, that allowed adult lactase production. This was found using genome-wide association studies, which we talked about earlier. Again, there we see evidence for a selective sweep in Africa too. And the estimate is selective advantage of about 5%. That basically the fitness of not having this mutation, the relative fitness would be 95% of that with the mutation. Again, these are concepts that we talked about. Now, this later date, this 5,000 years ago instead of 7,500 years ago, coincides with later cattle domestication in East Africa than in Europe. So again, we see a lot of the things that we discussed in class being applied for understanding human history. But in fact, the story gets even more complicated. That if you look in the Middle East, there is yet another origin of lactase persistence. And there it seems like it's the effect of two mutations together. You have to have both of them to get it. By requiring both, this is an example of epistasis, something we talked about long ago. There's evidence in this case that this may have been a response to camel milk consumption rather than cow's milk consumption. And these are probably not the only lactase persistence mutations out there. But this is really cool because we're really understanding a lot about ourselves, and we have these independent evolutionary events that are very similar happening in different human populations. Now, well, let me get you to apply something that you've learned here with a quick question. Given that these mutations are happened at different times and happened at different spots in the genome, what might you predict to be true from the following? So again, since we see these different mutations, they arose at different times, what we can infer is the most likely answer in this case is that there was a lack of gene flow among these populations. If there had been gene flow from Europe into these other populations, then there would never have been the need for this other mutation. It probably just would have spread in Africa, in the Middle East, etc. So this is again, migration or gene flow is a great homogenizing force of evolution, and again, more applications of what we learned. So putting this all together, we have these evolutionary genetic studies of an interesting condition in humans. This is a medical condition, I guess you could call it. It was identified by genetic mapping. This was what was used to isolate the cause alleles, and they, in doing this, identified multiple independent origins. Evolutionary analysis identified the strength of selection. Basically, we saw what the relative fitness was, and we see evidence of hitchhiking in molecular evolution in the genome. And this is coupling genetic data with evolutionary analysis to date the new mutation. And they inferred the cause from anthropological data. So this is tying everything together. So overall, again, these evolutionary insights that we've been talking about in the class can be applied to either aid humanity or understand humanity. And again, it's not all just math and dinosaurs. In the next video, we'll talk about some misapplications of evolutionary insights. I hope you'll join us. Thank you. Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about the application of evolutionary insights to aid humanity or to help understand humanity. In this video, we'll be talking about some misapplications of evolutionary insights. Times when people were perhaps good intentioned, but applied evolutionary thinking in ways that were not intended or that were not beneficial to humanity. So let's dive right in. Obviously, some mistakes have been made, and again, often by people who were well-intentioned, but what they did ended up having a very negative impact on humanity or on our planet. One classic example of people refer to is that of the introduction of the cane toad to Australia. This was a very intentional introduction by humans. I believe it started in something like the 1930s, where they brought over these cane toads to fight beetle pests that were found there at the time. Unfortunately, they didn't consider the biology enough, and there are no good predators of those cane toads found in Australia. As a result, they spread dramatically. They're found now across a huge swath of the country, or of the continent, really, and they've knocked out a lot of the native fauna there. So this has been a very bad thing ecologically. And again, it was well-intentioned, but it wasn't completely thought through. A more extreme example, potentially, this is a case where the biology wasn't considered at all, was again the intentional introduction of the European starling to the US. This was started with like 100 birds or so. It was not tons and tons of birds. One person released them to let people experience these birds that Shakespeare referred to in England, and when he was in England, but they released them in New York. Now they're found all over the continent. They have completely spread across the entire range. And they've, again, displaced a lot of the native fauna. This was a case of not considering the biology at all. Now, these are cases involving various animals, but what people sometimes do is they apply evolutionary concepts to justify other agendas, which may be somewhat darker. Now, again, in these cases, the evolutionary concepts in and of themselves aren't wrong, but the applications are wrong or at best questionable. So we see here in this particular picture I show, this was something from Cincinnati saying the feeble-minded or the hub of our wheel to vice, crime, and pauperism. Again, suggesting some sort of stopping of the feeble-minded or those who are labeled feeble-minded. Let's get into this, and we'll discuss both social Darwinism, as it's sometimes inappropriately called, and eugenics. Now, social Darwinism was popularized by Francis Galton, who was actually Darwin's cousin. And he pointed out correctly that a lot of mental traits are inherited, so things like IQ have a high heritability and various other talents. But what he then suggested from this, which is not as good, is that heredity should have a conscious decision. That essentially we should consciously allow those who we deem to be the more fit individuals to breed more than the less fit individuals. Now, in this case, we're not necessarily referring to fitness in the context of ability to produce a lot of offspring, but more fit in terms of how we think humanity should be. So, in this context, people on welfare or insane asylums may be considered less fit. So, the idea would be to limit their access to medicine or limit their ability to reproduce. Now, this was not popularly received, and even Darwin himself was against it. This is why I think it's inappropriately called social Darwinism. And Darwin responded to this, pointing out that some elimination of the worst dispositions is always in progress, even in the most civilized or advanced nations. That, again, a lot of criminals are executed or jailed, which limits their ability to reproduce, just in and of itself. Insane individuals are often confined or maybe suicidal, which, again, limits their ability to reproduce. And this was a great quote: "Violent and quarrelsome men often come to a bloody end," which clearly would limit their ability to reproduce. But he very strongly argued against any, what he referred to, check on sympathy. So not anything where we actually try to prevent particular people from breeding or hold back medicine or anything like that. And he says expecting the weak to not marry just won't happen. Nonetheless, this idea of eugenics was on the rise around these times, and this continued well into the 20th century. So eugenics is the attempt to improve the human race by eliminating those deemed defective from the gene pool. Now, this is very questionable, but nonetheless it was applied quite a bit. So I'll give you a couple of examples. In 1924, the Commonwealth of Virginia, which is one of the states in the United States, adopted a statute authorizing compulsory, so mandatory, sterilization of the mentally retarded. That's just amazing. I was personally shocked. I had no idea this was true. And in fact, this statute was not repealed until 1974, well into my lifetime. So again, this kind of thing is in our history, and this is a misapplication of evolutionary genetic insights. There was a famous case. That was the case of Buck versus Bell. So Buck was named for Carrie Buck, who was a Virginia inmate in a mental institution. Her picture is there on the screen. And she was to be sterilized for, quote, immorality. This went all the way to the Supreme Court, and in fact, the legendary Oliver Wendell Holmes read the statement on it, and I have it quoted here. "It is better for all the world if instead of waiting to execute degenerate offspring for crime, or to let them starve for their imbecility, society can prevent those who are manifestly unfit from continuing their kind. 3 generations of imbeciles are enough." So they upheld it, and I believe Carrie Buck was in fact sterilized. Nonetheless, she actually did have a grandchild, and her grandchild was actually in the honor roll at school, which shows that, again, genetics doesn't explain everything. But again, these ideas were very abundant around this time. And Ronald Fisher, who is a famous statistician and evolutionary biologist, advocated this. He reiterated this hereditary basis for offspring number and for temperamental qualities. Now, this is very similar to what Francis Galton was saying. And he pointed out the birth rate is much higher in the poorer than in the more prosperous classes. And I have there a picture showing the correlation of the size of mother's family on the x-axis with size of daughter's family on the y-axis, showing that, generally speaking, people who came from large families tend to have large families. And it tends to be, according to him, the poorer that have more kids. So what he suggested, and this is again the questionable area, is the redistribution of births would be attended by economic advantages. That essentially that the more prosperous should be allowed to have more kids, and in some way we should hold back reproduction from the poorer classes. That this redistribution should favor the more successful. These are all very subjective qualities. The more successful, the more fit, all these kinds of things. Now, the thing that a lot of opponents of evolution always bring up is this idea of Nazi Germany adopting this sort of thing. There was a Nazi euthanasia program that was instituted to eliminate, quote, life unworthy of life. It's a very awful way of putting it. And in fact, they saw people with disabilities as a drain on resources, and many of them were killed in huge numbers, as shown in this awful picture here. Now, I want to stress, because this often comes up, and people say, oh, see, evolution is evil, Darwin was evil. In fact, contrary to claims, Hitler was not influenced by Darwin or by true evolutionary theory. There's no reference to Darwin whatsoever in Mein Kampf, even though he was advocating this. Darwin, as I mentioned, disapproved of eugenics. So this is not following Darwin. This is going against Darwin. And in fact, Hitler believed in intelligent design. Really? So here's a direct quote. "For it was by the will of God that men were made of a certain bodily shape." That is very much intelligent design. That is not the standard theories of evolution. So Although people sometimes throw this out as trying to tie Nazi Germany evil with evolutionary biology, it's not true. Now, let's take it a step back. Why is eugenics wrong? I've been sort of implying through this whole time that eugenics is wrong, or at best questionable, but I think it actually is wrong. Well, again, as Darwin said, selection is still operating on humans no matter what. That we do have bad mutations coming up all the time and being eliminated from the population just naturally without any human interference. The heritability of bad traits is not always very high. So taking individuals and trying to stop their reproduction because they have these bad traits will not necessarily change how much these bad traits are apparent in the next generation. The other thing this does is this reduces variation that may be helpful in case of a change in environment. So if you go back, say 500 years ago, somebody like Bill Gates or Steve Jobs, who have been extraordinarily influential and successful by many measures, would not have necessarily been considered even above average. They may have just been like, oh, there's this, skinny person who's kind of smart. Finally, and probably most importantly, who's to decide what's best? Who's to decide what is most fit or what is considered most successful? Is it just by money? That would be ridiculous. Fundamentally, dictating who can marry or reproduce is considered by most people to be immoral. And therein lies the problem with eugenics. But ultimately, there is no inherent good or evil in any sort of science, really. We can apply it for good, as illustrated by the previous video, or we can apply it in poor ways, as was illustrated by some of the examples in this video. It's nonetheless incumbent upon us as scientists and as citizens to consider the consequences or ramifications of how we interpret the science and to take very great care in how we present it. And ultimately, we all have to behave morally. Well, thank you very much. I appreciate your time.